Good morning, students. Today we'll study chapter number 2, that is sexual reproduction in flowering plants. All flowering plants, they belong to division angiosperms. And as you studied in 11th standard, angiosperms' dominant generation is sporophyte, means they produce spores. They produce two different type of spores. That's why they are called heterosporous. The larger spore is called megaspore and the smaller one as microspore, which later on forms the male and female gametophyte, which produces male and female gamete for sexual reproduction. In angiosperms, flower is the site of sexual reproduction. It is modified shoot. A flower is attached to the stem of a plant by means of a stalk that is known as pedicel. The tip of the pedicel expands and it is known as receptacle or thalamus. On thalamus, 4 floral holes are arranged. The outermost floral hole is known as calyx and its individual member is known as sepal. Inner to that is present corolla and its individual member is known as petals. The third floral hole is androecium and the individual member of androecium is known as stamen. And the innermost floral hole is known as this one, gynoecium, and individual member of gynoecium is known as pistil. So we can classify these floral holes into two categories, non-essential floral holes and essential floral holes. Non-essential floral holes, they are not involved in sexual reproduction. Okay, and they are calyx and corolla. Essential floral holes consist of male reproductive structure known as androecium and female reproductive structure known as gynoecium. Androecium and gynoecium initially produces spores and these spores later on forms the gametophyte in which the formation of male and female gamete takes place. So this is stamen. A stamen consists of two parts. One is anther and attached to the anther there is present a long tube known as filament. Okay, anther is filled with pollen grain. It is bilobed having 2 lobes and it is filled with pollen grain. Then in the center pistil is there. Flower may have one or more than one pistil. Accordingly, it is called monocarpellary or polycarpellary. Okay. So a single pistil, if you study, it consists of 3 parts. That is stigma. It is the sticky receptive surface for the germination of pollen grain. Then there is a long tube that is known as style and at the base of the style is a swollen part present known as ovary. Inside the ovary, there are present one or more ovules. This structure, green colored, is the ovule. And inside the ovule, the cells, they undergo division and finally they lead to the formation of female gametophyte. So, this is female gametophyte and inside female gametophyte, female gamete, that is egg cell, is formed. Okay, so this whole structure is known as pistil or carpel, which is the female reproductive part. Now we'll study the pre-fertilization events and structures in detail. First, we'll study about the androecium. So we studied that androecium consists of holes of stamen. And a stamen, it consists of 2 parts, a long slender stalk known as filament, which is attached to the base of the thalamus or it may be attached to the petal. And at the tip of the filament is present bilobed anther. Okay, this is anther. It is having 2 lobes. Okay, and inside each lobe there are present 2 pollen sacs, which are also known as microsporangia. So in each lobe there are 2 microsporangia. So total 4 microsporangia are there inside an anther, bilobed anther. These 4 microsporangias, they are located at 4 corners of the anther. Okay, and they form microsporangia, which later on matures into pollen grain. That's why microsporangia is also known as pollen sac. Okay, now we'll study about the structure of microsporangium. Okay, so this is a single microsporangium. The first diagram is the diagram of transverse section of mature, or you can say young anther. So a mature anther, as we studied, it is bilobed and it consists of 2 microsporangia. Total 4 microsporangia at 4 corners, and the 2 lobes are connected with each other by means of a tissue known as connective. Now, if we take a small section of one of the microsporangia and we observe its enlarged view. Okay, so this is the enlarged view of microsporangia. Okay, each microsporangia is covered with 4 wall layers. The outermost wall layer, this is epidermis. Inner to the epidermis is present the next layer known as endothecium. Inner to endothecium, there are present 1 or 2 cell layers known as middle layers. So these 3 outer wall layers of a microsporangia, they are protective in function. They protects the developing microspore, and as well as they help in dehiscence of the anther. Now, so these are the 2 function of outer 3 layers. Okay, one is to give protection to the developing microspore, and second, once the microspore matures into pollen grain, it helps in dehiscence of the anther. Now, the 4th wall layer, it is named as tapetum. This tapetum layer, its function is to provide nutrition to the developing pollen grain. Okay, so here in this figure also you can see the outermost layer, this one is the epidermis, then inner to that is endothecium, then here the middle layers, and this one is the tapetum, and in the center the space is there where the spores are formed, microspores are formed. So tapetum provides nutrition to the developing microspore. In the center, okay, of each microsporangia, the tissue which is present here, the tissue which is present is known as sporogenous tissue. So sporogenous tissue forms the center of each microsporangia. The cells of sporogenous tissue, they divide to form microspores. Okay, that's why these cells are known as microspore mother cell. They are also known as pollen mother cell because microspore, when matures, it forms pollen grain. Okay, so microspore mother cell, we can also call them as pollen mother cell or microsporocyte, or we can also call them as meiocyte as they divide by meiosis. Okay, cells of tapetum, it consists of dense cytoplasm and they are cenocytic, have multiple nuclei in them.